Okay, hi everybody. We're back for part 2 of the urinary system. Part 1, we talked about filtration. So part 2, we're going to talk about tubular reabsorption. So if you recall that at the Bowman's capsule, we filter the blood from the glomerulus. So the fluid entering the Bowman's capsule now is called the filtrate. And we mentioned we don't really seem to have the ability to control what gets filtered. So we filter a lot of stuff we don't want to lose. That's where the process of reabsorption comes back in. So reabsorption, we're transferring substances from the lumen of the nephron into the peritubular capillaries. Okay, this is a highly selective process, so that should tell you this is no simple diffusion. We're going to have transporters for specific things to move back in. Okay, and it says here it involves transepithelial transport. Remember, epithelial cells are what make this tube. So in order for anything to leave the tube and get into the capillary, they have to cross the apical surface of the cell, the basal surface of the cell, into the interstitial fluid, and then cross into the capillary. Right? What do I mean? Here's a close-up of that. So here's a nice, the nephron. Here it's showing you these are simple cuboidal cells that line the proximal convoluted tubule. So this is a step that happens in the proximal convoluted tubule. Okay, and so this is what you see here. This is a blow up. Here are the cells. We have tight junctions in between the cells. So the fluid, the filtrate, can't leak between the cells and get in. The substance has to move from, again, the apical surface all the way across. It's the reabsorbed substance is actually crossing 5 barriers. That's what these numbers are for. Okay, so this shows you what they are. The luminal surface of the cell membrane, it's got to cross this one, or otherwise known as the apical surface. It's got to cross through the cytosol, or the cytoplasm. All right, that's not going to be a big deal. Then it's got to pass through the other side of the cell membrane, either so the basal or the basolateral surface. Okay, it's got to go through the interstitial fluid and then cross the capillary wall, which actually has 2 surfaces as well, but we're not going to worry about that. Okay, so again, these are the steps of what's called transepithelial transport. Trans means going across what? The epithelium, right? And we're transporting it. Okay, so again, this is all occurring at the proximal convoluted tubule, at this portion, up in the cortex. All the way around here, that's where this is happening. Okay, so at the proximal convoluted tubule, the function we get there is reabsorption. Okay, and there's going to be 2 types as we go through those different barriers. Sometimes we'll get passive reabsorption. Okay, no energy is required, but it's going to cross through a channel. Okay, sometimes we're going to need energy, and so that's going to be active reabsorption. Okay, so we might have to move the substance across against an electrical gradient or a chemical gradient. Okay, so remember passive and active. Okay, and the same substance might move on one side of the barrier through passive reabsorption, and the other side it might have to use active reabsorption. Okay, all right, that's what we see here. Let's do sodium. The proximal convoluted tubule reabsorbs sodium. Okay, because we just we filter too much sodium out. We have to reclaim some of that. So inside the tubule, sodium is highly concentrated because we just filtered a lot of it out and put it into this tubule, filtered it out of the blood, right? So it can't cross the membrane, so there are actually channels in there, and they're open. So the green is showing you passive transport, right? Passive movement of the ion down its concentration gradient. So it does. It can pass the membrane because it's got an open channel, but it's just going with its concentration gradient. So there's no energy required here. Now it's in the cell. Okay. Now there's actually more sodium in the interstitial fluid and in the capillary than there is inside of the cell. Okay, remember that's just true. Remember, go back to your neurons. Whenever we opened a channel, sodium came into the cell because there's always, just by definition, there's always sodium, more sodium in the interstitial fluid and in the blood. Okay, so to get it out of the cell, to pass on the other membrane, the basolateral membrane, it's got to go through a sodium-potassium pump, ATP, primary active transport. We're going to pump sodium out, and any potassium that's in here, we're going to pump it in. Okay, so there you go. We expend energy to do this. So the total of the total energy spent by the kidneys, 80% is used for sodium transport. Okay, sodium is not reabsorbed in the descending limb of the loop of Henle. All right, it's mainly going to be reabsorbed here. Okay, so water also will follow this reabsorbed sodium by osmosis. So the PCT also reabsorbs water. So we just did two things. We reabsorbed sodium and we reabsorbed water. Okay, because we made it hypertonic out here, right? So when sodium goes, water's going to follow. Now you have to always remember that this fluid is constantly moving down the tube. Pretend like you're always sucking on your straw. So fluid is constantly moving through. Okay, so as water moves out, then it becomes a little less dilute down here. All righty, so by reabsorbing sodium, we do it passively by facilitated diffusion at the luminal surface or the apical surface. And then we do it with primary active transport with the sodium-potassium pump at the basal surface. When we get sodium to move across, it enters peritubular capillary passively, green. Okay. Water follows. All righty. The other thing that it reabsorbs a lot, but I just want to go over sodium, water, and the third one now is glucose, right? Because glucose is so important. We filter glucose out, but we need glucose to make ATP. We're using ATP now, right? So we can't afford to lose glucose. So we reabsorb everything. Okay, a healthy kidney reabsorbs the kidney. Okay, and so it's going to do this through a secondary active transport because we have a lot of glucose inside of all of our cells because, again, we're making ATP. So there's mitochondria in here and they're using the glucose. But this cell of the PCT, they have to take the glucose that got filtered out and put it back into the blood. Okay, so it's going from the area of even though there's a high concentration, it's lower than what's inside the cytoplasm. So we need active transport, but we're not going to do it primary. We're not going to expend ATP here. We're going to use the fact that sodium transports across. Okay, so sodium, again, is going to go through its transporter, and this transporter is called a sodium glute transporter. So that means it's a co-transporter. Okay, remember transporters are specific. So this is specific to sodium and glucose. When sodium moves through passively, right, that creates a current. Remember, this was the definition of our secondary active transport. It creates a current so that as sodium moves through, glucose is specific to this transporter. So it can bind and then the sodium helps to pop it across against its concentration gradient. Okay. So it's doing that against its concentration gradient. Alrighty, and then once it gets inside the cell and has to move into the interstitial fluid, well, it's a lower concentration out here. So that's just facilitated diffusion. Okay, facilitated diffusion. I'm sorry, I'm reading something. I want to see if I want to mention this or not. Yeah, we're not gonna worry about it. All right, so I just want to stick with, so what's the method of moving glucose across? So make sure you understand secondary active transport. Rewind this part if you need to. So we saw the PCT reabsorbs sodium, water follows, with the reabsorption of sodium in our GLUT transporters, we can get glucose to move across against its concentration gradient into the PCT cell, but then with its concentration gradient toward the capillary. Okay, I'm trying to advance. I can't advance. There we go. Okay, so basically, the renal handling of glucose, this is telling us about what's called a tubular maximum. Okay, so Tmax. This means this is the amount of glucose that our kidneys can handle reabsorbing. Okay, and it's a finite amount because there's only a certain number of these transporters, right? So if these transporters all get busy, and now imagine they're all along these cell membranes, right? So if sodium and glucose are busy on this transporter, that means the next glucose coming along can't bind to this one. It's got to move to the next transporter that might be available. If that one's taken up, it's got to move to the next one and the next one. So that's what this means about the transport maximum. There's only a certain number of transporters, so there's only a certain amount of glucose that we can transport across and reabsorb. Okay, when we have so much glucose that we can't, if we have so much glucose in our blood vessels and we filter so much of it, we won't be able to reabsorb it. It's actually going to get excreted. So there's a maximum of how much we can reabsorb. So the average Tmax is about 375 milligrams per minute. That's actually a lot higher than the rate of glucose that's normally delivered. So in a healthy body, the plasma delivers or gets filtered about 125 milligrams of glucose per minute. So under normal conditions, all the glucose that gets filtered gets reabsorbed. Okay, because we have enough transporters to handle that amount of glucose. Okay, but glucose plasma levels are maintained mainly by the pancreas and the liver mechanisms, right? So if you have too much glucose in your bloodstream, maybe if you're diabetic and you're not releasing enough insulin so that your other cells take it up, right? Then you're going to have more glucose in your plasma, and more than, if you get more than 375 milligrams per minute, right, being filtered. This is the stuff that doesn't get reabsorbed, right? And therefore it will get excreted. Okay, so the renal threshold is about 300 of our plasma concentration. That's the concentration. 375 is the rate, milligrams per minute. Okay, so our normal blood plasma can hold up to 300 milligrams per 100 milliliters. Okay, so anything above that gets excreted out. So again, how do you remember this? T maximum, I think of that as the number of co-transporters that are available. So the numbers we have can only handle about, what I say, 375 milligrams per minute. If you have more glucose running through here than that, then it passes through the PCT because all these doors are open, or all these doors are clogged up, right? So again, if this is unavailable, then this glucose is going to move to the next one, move to the next one. If it gets all the way out of the PCT, we don't have any more chances of reabsorbing glucose because we don't have the transporters on the other part of the nephron. Okay, I always relate this to like when the good old days when you could go to a concert. That wasn't too long ago, right? And at the end of the concert, everybody's emptying out of the room into the hallway, and there's always a lot of doors along the hallway leading to the outside. And you know your car is over here, but so you're trying to get out this door. No, it's clogged, too many people. And so you try to get out this door. No, it's clogged, too many people. And you're trying to get out this door, right? And you're in the flow of people, and it just keeps pushing you down and down until you finally get out a door way at the end, and you got to walk all the way back to your car. Right, so I think of those doors, there's a limited number of doors. That's like the limited number of these co-transporters, right? That's Tmax. Okay, So that's reabsorption of glucose and renal handling. All right. Let's see. So when we reabsorb sodium, remember it's passive on one side, but then it's active on the other side. Chloride follows. That makes the outside salty, right? So sodium or water follows, right? I mentioned glucose. We also actually reabsorb some urea. Urea is a waste product of protein. And so of the nitrogenous waste I told you about. So as water is reabsorbed, urea becomes more concentrated in the tubule, and that sets up a concentration gradient with the capillaries. So about 50% of it gets reabsorbed. And we can handle that. It's not very toxic. So we convert the nitrogenous waste into a less toxic form. So sometimes it actually helps with osmolarity of the blood. But mainly if you can tell me about the reabsorption of sodium and water and glucose, you're good to go. All righty. That's going to bring us to the loop of Henle. So I am going to stop the video here and do a new one for the loop of Henle. So we'll be back.